All right, with all that half-hour preamble, let's get into the journal club. Probably another half hour. And I also do want to, this is going to be a little bit of a longer one because it's a complex paper, it's a complex technique, and it's also going to involve a couple other videos for you to watch, but don't worry, only one of them is longer than 2 minutes. Okay? So this is our fourth journal club, and we're going to talk about, I want to show you the highlights of the things we're going to talk about. We're targeting cancer with antibodies. Look at those words. Now, it's a particular kind of cancer that's RAS-driven human cancer, and it's cells. We're targeting them with antibodies to upregulated and essential cell surface proteins. Well, that's interesting because cancer is by definition inside the cell. It doesn't make many changes to the outside of the cell, and yet the outside of the cell is much easier to reach. So if you want to fight cancer, I suggest that we do papers like this. This paper is pretty amazing and pretty complex. It involves a process called the antibot. And it's important background because we're going to talk about the antibot. How does it make so many antibodies? We just talked about how the body shuffles genes and then does somatic hypermutation to make a lot of antibodies. Well, this does it, but it does it like a robot will. And robots always do things different than life will. This robot is basically really big on somatic hypermutation. And so I want to show you what that means as we go through. And we're also going to talk about the paper. How can you use immunology to fight cancer? It's called immunotherapy, and it's basically making antibodies that will specifically bind cancer cells but not regular cells. And this sounds like something your immune system does every day, and at some level it actually does do this every day. But the trick is that there are a lot of forms of cancer that are only changed internally. Antibodies can't look internally, and so they can only bind stuff on the outside. And if there's no outside changes, there's nothing for an antibody to bind. Or if it does bind, then it's binding all your cells and you have an autoimmune disease. So we want to have immune systems to fight cancer, but one of the tricky parts is finding that external protein that actually will indicate that it's always there when there's cancer. It's never there when there's not. That's a really hard thing to do. So there are a couple other videos associated with this lecture. I just recorded an intro to this journal club. Make sure you watch that. And then there's a very short antibot video. Make sure your volume's not turned up too loud. You'll know what I mean when you actually see it. But do listen to it. It's fun. And it's very short, but I want you maybe to watch it a couple of times and think about where have you seen stuff like this in the lab that you've done before. The paper is also open access, so I encourage you to actually have the paper. I like to print them out. I know it costs money, but it's so helpful to be able to look back and forth and write on it. You remember things when you write them down. So I encourage you to have the paper at least accessible. I'm going to refer to it because I want you to learn how to read a paper, even a paper as complex as this one. So in 2014, cancer immunotherapy was one of Science Magazine's Breakthrough of the Year, which means we get lots of nice pictures like this. You can see the antibodies right here. This is clearly some kind of FC receptor rather than a actual variable region. If you know enough about antibodies, you can kind of look and see if these are accurate pictures or not. And as you found out from our first test, it's not always accurate. Sometimes errors slip through, just like with DNA. But immunotherapy was really expensive even back in 2010. It was already surging, and you can see that individual drugs, there's this Yervoy drug that is an immunotherapy, it's really expensive. And it's only gone up from there. I should probably look up a recent graph, but I don't think you need convincing that medicine has gotten more expensive. So this is actually part of a problem, but it's a topic for another class. Here we're going to talk about why it's so expensive. But you're going to have to think about, is it worth it? You know, is it effective enough to be worth the cost? And we really have to ask that question because the prices are continuing to go up. It's not because scientists are greedy. It's because scientists, the science that we need to do is really complicated, including the antibot. Now, hopefully you've been able to watch the antibot and you've seen it move around, maybe identified a few of the things like 96 well plates, buffers, even the bottles that we use, like the media bottles. If you can see that we have the same orange lids, you know, once you use those here, you can use those anywhere. And you will use those anywhere. And it's located at UCSF. This is where there's a scientist named James A. Wells, who I actually have been following for a long time. I first knew him as protein design, where he used phage display to design a small little protein to ask really interesting questions. I use his paper in biochemistry sometimes, and then it looks like he's moved on from designing just small proteins to designing antibody fragments. And this is the antibody, and it's not really designing antibody fragments. It's doing it the same way B cells do. It's making a bunch of different DNA sequences and then throwing them at the binding surface and seeing what sticks. This is what the website is. This was current a couple years ago. I think it's still up. They have actually antibodies at multiple places, and there are recombinant antibody networks where they will actually, I think you can hire out some of these services. Yeah, you see there's U Chicago, U Toronto, and UCSF. I think sometimes they serve the researchers they work with. And there are some places that have antibody generation for hire, although I'm not clear how much of it is in animals versus how much of it is robotics. I do like this from an ethical standpoint because it doesn't have any of the problem of destroying life to save life that you have when you are destroying an animal in order to get their antibodies. Ethically speaking, we do have an equation to solve when we have a child dying from brain cancer versus an animal, but I do like the fact about the antibot that it doesn't have that ethical dilemma, and it's biochemical in nature. So this is the paper, and this is what I want you to look at. We already talked about how it's about targeting cancer. RAS-driven cancer cells. We're going to start with that, and then we're going to get to, it doesn't actually have antibot. I don't think it has it in the whole text. So I want to ask you to look at the abstract and answer these two questions. Which sentence was done by the antibot, and what's the overall purpose of the study? Remember, the antibot will generate antibodies. So look for these two sentences. I'll let you pause and look at it for a second. Okay, here's your answer. The antibot is shown in this sequence. We generated a toolkit of recombinant antibodies to 7 of these RAS-induced proteins. In other words, they used the antibot to make 7 antibodies, and they then used those to study cancer cell lines. And what's the purpose? This work presents a technological platform for attacking RAS from outside the cell. And that's really impressive if you remember, RAS is an internal signaling protein that's mutated in cancer. The thing that you see RAS above, look above, and you see KRAS G12V. That's a particular mutation. It's from the amino acid glycine to the amino acid valine. It's a single mutation that turns a cell into a tumor cell. This is why RAS is a central protein for cancer. There's a lot of mutations to it. It is so important to cell growth that it can be mutated very easily to accidentally be left on. The G12V mutation will be one of these mutations. And in fact, that the G12V mutation, if you just put that RAS into a cell, it will make that cell into a cancer cell. And so, if you look on the right, you have the cancer cell with the G12V. If you look on the left, you have, I'm sorry, I said that wrong. On the left is the G12V KRAS. On the right is a normal cell. And you don't need to be a biologist to really look at those and say, oh, if the cell on the right is normal, then it's pretty clear the cell on the left is abnormal. This actually gives us some hope. If it looks so different from this view, maybe there's some proteins that are different on the surface. There's a little bit of hope, and we found some proteins. Only found 7. And even of those, only 2 worked. And of those, only 1 really worked well. So there's a lot of back and forth. Oh, great, we're so back. And then we're so, what's the thing that we're so back? We're so done? Is that the phrase? Anyways, the whole we're so back, we're so done kind of thing. That's a thing about all of life. So an antibody that binds a particular receptor on tumor cells will make a cancer treatment. It turns out that there are some forms of cancer cell, breast cancer, that express more of a particular receptor called HER2. And so those are called HER2 positive breast cancer cells. And Genentech is a company that made an antibody that would bind two of those cells, would cross-link them, and when you stick two of them together, it turns out that they stop signaling the cell to grow. So having too many of these receptors is what makes the cell a cancer cell. So this is the perfect starter cancer thing for a company like Genentech. You make an antibody to an external signal, you can recognize and bind these receptors, and just by binding them together, it stops them from signaling the growth signal, which is really, really kind of cool. So James Wells worked for Genentech. He started out at UCSF, then he worked for Genentech, and somehow he got back to UCSF again. I don't know how it works, but these scientists, they start companies and they do stuff because the biotech can help a lot of people. It's also expensive. So there's our dilemma. Genentech was founded on the success of Herceptin, and now we can look at Figure 5. So if you have the paper in front of you, turn to Figure 5 and look at those antibodies. Ask how are antibodies used there. Immunology folks, remember that we said that Herceptin is the trademark name. The official name ends in mAb, and that tells you it's a monoclonal antibody. One of the things that James Wells did is he actually designed the antibody to be humanized. More biochemistry than we can talk, but we can tell that it was a humanized antibody by looking at its official name. Its generic name will actually say, I can decode it, and we can see what kind of antibody it is. Those are really cool. I might give you that table and ask you to decode an antibody name for me, but I would not ask you to memorize the table. All right, so Figure 5, this is what it looks like. And it shows how antibodies can be used. There are two places where you see antibodies in this. They show it to target and detect RAS signaling from the outside of a cell. And once you do that, you can see that they're using it particularly for pancreatic ductal adenocarcinoma. Okay, so that tells you something about what their end result is. Look at the antibody and see it's got the alpha, which means anti, and the thing after the alpha tells you what it binds. CDCP1 shows up in both of these. This is a CDCP1 binding, so the little blue protein on the tumor cell is a CDCP1 protein. It's one of the things that they found that differs on tumor cells. You find it on tumor cells and not on normal cells, and in fact it's particularly pronounced in pancreatic ductal adenocarcinoma. That's why they're going after it, not because they have a grant to look at that, but it's because they found it's the best example of CDCP1 tumor-associated expression. Once you have this blue antibody, you can do two things to it. You can basically stick the antibody to a radioactive bit of nucleus, something cytotoxic, something really nasty. And what you do is you make, for technical reasons, because it's easier to make FC binding antibodies than it is to make antibodies in general, what you do is you make the blue antibody, which is the one that binds the tumor cell. You see it's binding the tumor cell right there. Then you make a secondary antibody. So the blue antibody is the primary antibody, primarily binding the cell. Then you make a second antibody that's a deadly antibody. And what it does is it binds the FC region of the first antibody. This is just so you don't have to put the nasty stuff on your precious primary antibody, but instead you can make the secondary antibody in bulk. Also, the primary antibody can have some differences in the FC region that allow the pink antibody to bind only it. Once you do this, you make sort of a daisy chain of interactions. And the blue binds, the blue binds, and then the red binds, and the red has a poison on it, and that means that the poison is right next to the tumor cell, and it gets into the tumor cell because it's sort of targeted to it, kills a few other cells, but you do safety tests to see, hopefully, that it's not killing too many other cells. There's a more immunological way of doing things where they take not a full antibody, but an antibody fragment, just the FV fragment. Remember, it's just 4 blue domains there. It's still binding a tumor cell, but attached to this is going to be a single chain FV. Remember how we said that you could actually have different smaller versions of antibodies? This is one where they've taken a single chain with just 2 domains and 1 binding site. And this is something that binds CD3, which is a cell marker on T cells. In fact, it's on cytotoxic T cells. And so what happens is that you have the blue end of the antibody binding the tumor cell. The red end of the antibody binds the green T cell. In this case, they just made it glow green so they could show that it would work, that it's activated. But in the real life situation, you could have it go for a T killer cell that would then destroy the tumor cell. So either way, you're bringing something nasty close to the tumor. On the left, you're bringing a poison close to the tumor with a secondary antibody that is non-covalently attached. It binds to the FC region. On the right, you are using a fused protein that will actually literally bind with one end. It's a two-ended protein. It'll bind the tumor cell with one end. It'll bind CD3 on T cells with another end, and that will bring them close and activate the T cell. No more tumor. That's the idea, at least. There's another purpose for this, which is just imaging. Having a good imaging reagent is worth a lot, and they showed that this can work for the antibody that they made. So they showed this has a practical therapeutic use, which a lot of papers aren't able to show. So they showed this can image tumors in mice. They used a PET imaging study, and basically on this, the tumor shows up red. How much red do you see, and how specifically is it bound to the tumor? And what they do is they have a FAB antibody fragment that binds CDCP1, just like on the other things. And instead of being bound to a poison, it's bound to a PET probe. In this case, it's a particular isotope of zinc. And you give that to a mouse with a tumor, and you see how well it goes to the tumor and how much it lights up. And here they show it. It's lighting up very specifically. This is a mouse with a tumor in the middle, and you can even see the shape of the tumor. It looks like this is working very well as an imaging reagent. And if it works in mice, it could work very well in humans as well. If you know where the tumor is, that's part of the problem a lot of the times, finding out where the tumors are in the body. So, whenever you're reading a paper like this, you want to scan through the papers, you want to see what's their purpose. Their purpose was to make antibodies. We just talked about how the antibody that they found was for that purpose, and we also saw which their protein was that they ended up with. You do want to start at the end. Spoilers are good in science. And so, flip to the end of the paper, flip to the last section, and try to look at this section to see the section with the title at the top, and try to look for the name of that protein we were just talking about. You want to say that's the focus of the whole paper. So now I will unpause and show you. There you see CDCP1 several times in that paragraph. That's what you want to see. They chose that. They showed that it works practically in the lab as one of those three different kinds of ways of using things. They showed it worked as an imaging reagent, and they speculated that it could work as the other kinds of reagent. I think they did some experiments like that. But we want to look at the antibody. And so you want to see that CDCP1 looks like it actually works. That's great. But then you might ask, you were scanning literally all the proteins on the surface of the cell, and you just end up with one? So let's talk about all the effort that they went through. They screened the entire surfaceome of the proteins. Everything, the genome is all the genes, proteome is all the proteins inside. This is all the proteins on the surface, so they called it the surfaceome. It's one of those things that was really big about 10 years ago, calling everything omics. So the surfaceome, the way that they screened is they actually used mass spec. We won't get into the details here, except I want you to see that there were actually two directions of experiment, and both of them gave mass spec signals. The thing about mass spec is really cool. It weighs every single protein, but you need some way to discriminate the proteins to be able to tell which protein you're looking at. And how much there is. To tell how much there is, you definitely need what's called an internal standard. Something that's exactly the same, but it's different in a way that won't change the chemistry at all. The internal standard they use is they use different isotopes of carbon and nitrogen so that they have different masses, but the isotopes don't change the chemistry at all. This is a great sort of internal standard. It's the same kind of thing that Dr. Pierce does when she does chromatography. It's just done with proteomics instead. We're not going to get too much into how and why they use it, but just know that they use mass spec, and they need to use isotopes to be able to measure how many proteins they have, and they're always comparing one state to another. Notice that there are two different comparisons done here. On the left, you have the regular protein versus the cancer protein. The regular protein's in red, and it's got light isotopes. The cancer protein's in blue, and it's got heavy isotopes. You actually can culture those cells together, you mix them up, you put them through the mass spec, and because you have both, you can tell light from heavy in the mass spec, you can tell how many you have of each protein, enough to say, oh, this protein is here because the cancer is here. Okay? But that's not enough. Cells are really complex. There's tons of proteins on them. There's another direction, and I think this is pretty brilliant. They also do a light medium and heavy medium, and both of these have the mutant genes, so both of these are cancer. But they also have specific RAS drugs that will turn off the RAS when it's left on by the mutation. It'll actually go and turn off the mutated RAS again. So it's like you're adding the cancer on the left and you're taking away the cancer on the right, using the drug to do that. Now, different drugs will do this slightly differently. It's complex. But for one drug right here, it shows you that what you do is you do without drug, red is cancer in this case, and blue is drug treated, so the cancer has been turned off by the drug. And then you see what's light and heavy. Hopefully what you see in that case, you're looking for proteins that have reverse effects when they're in blue on the left and in red on the right, or vice versa. Hopefully you look for something that is blue on the left because it's there when it's cancer, and it's not there when the cancer is turned off. So for each of the different kinds of proteins, this is the one where you just are comparing not regular RAS to mutated RAS. And so they're doing the blue and red thing, and they're measuring the expression levels, how much of each protein you have as measured by mass spec. And the farther up it is on this, and the farther away from zero, the more protein you have. And so you have a lot of noise at the bottom. But then you have some proteins that you definitely have more of. They're showing up on the top of the graph. And what you really want to do here is they've labeled the ones that they think are important. Other ones, they found out that they weren't important. But these ones show up in both blue and red, and they show up as upregulated in red and downregulated in blue. And then you have the converse. They found out when they added the drug, they went down or up in the opposite way. And so if you look at this, yeah, you do have these labeled cases. They have 13 surface proteins that they found when they did this with the MEKI inhibitor, okay? So and I've connected the dots for you. You can see where there's the label, and there's the label, the ITGB1 protein. You see that it's blue on the left and it's red on the right. That's what you want to see. That means that it could be something that's there when it's cancer and not there when the cancer is taken away. The CDCP1 is another one. And so you can see, oh, okay, that's the one that they found. So that can help you make sense of this. Then there's another bunch of them. You see that there's 13 that they found that showed the right kind of pattern. But there's a lot of issues. There's a lot of scatter. There's a lot of error in this data. And so you just start with these as candidates. You need to actually test them. So they do a big Venn diagram. They did this with the KRAS activation. That means cancer, no cancer. Then they did this with 3 different inhibitors. It makes this kind of big Venn diagram. But if you look at this, so you can see that when they saw changes on the surface, they tell you how many changes on the surface they saw. And they only saw, for example, they only saw 13 changes on the surface when you compared the MEKI data to the KRAS activation data. That's shown in blue right here. So look at that and see 10 plus 1 plus 2 adds up to 13. That's cool because that's in the area where the Venn diagram overlaps. You can look up and down. You can see one of those also showed up with AKTI, and 2 of those also showed up with EGFRI. None of them showed up with all the inhibitors, so see that there's a lot of zeros, and you can add them all up to see that the MEKI definitely produced the most surface changes. KRAS activation produced a lot of surface changes, so they focused on that because it was 13. It's a lot more to look at than if you look at the other ones. There's really only 2 in one case and 1 in the other case. There's just not as many changes. It looks like the whole system worked better with the MEKI. Why? It's because of the complexities of chemistry. We may not know, but we do know that we have 13 candidates, and it's better to chase after that than to chase after a bunch of other ones. The other thing that they did is they did, a lot of times you'll see this done with RNA data. In fact, we've seen this in the spleen paper, the blackened spleen paper, or not spleen, but lung lymph nodes. You know what I was talking about. That paper actually used mRNA expression to say what was on the surface of the cell. Well, that's not always reliable because their method here, called SILAC, remember it's based on mass spec, it's based on actually seeing the proteins on the surface of the cell. The RNA on the inside of the cell should go with it, right? Because the RNA is making protein, but not necessarily. And in fact, what they did is they compared RNA sequencing data to their SILAC technique. And they saw if they correlated. Basically, if they saw a lot less of the protein, it got red on SILAC. They saw a lot more of it, they got blue on SILAC. They saw a lot less of the RNA, it got red on RNA, and it got blue on RNA if they saw a lot more of it. And you see that at the ends, you have many cases where the red and blue are lining up. But look in the middle. There's a lot of cases where the SILAC gives a blue or white response, and the RNA-seq gives a red response, gives the opposite response. And they sort of organize these so that you can see the colors better. But there's about half the data, it does not go together. And so this is like, okay, mRNA is okay, but this is actually, one of the things they're showing is that this proteomics thing is going to give you some data that mRNA will not. There's not a correlation with mRNA. It doesn't mean we shouldn't do mRNA, but we need to have multiple techniques because sometimes mRNA levels will be high when the surface protein will never make it to the surface for some reason. Again, we don't know why. We just know that they don't correlate always. There's places where red on top and blue on the bottom, or blue on top and red on the bottom, and that means they disagree. So you can't just do RNA, and there's a place for this technique. Now this is the key of the paper. This is where they use a biochemical technique called phage display. If you've heard of bacteriophages, they're viruses that infect bacteria. You can use bacteria to make proteins. You can use these viruses to infect them. And the thing about the viruses contain genetic information. That means that the virus actually will carry the genetic information and can be sequenced so you can select out a particular protein and it will still be bound to the bacteriophage, which means you can sequence the genetic information in the bacteriophage and get the sequence of that protein that you're interested in. This is really cool. It's also really hard to do, and this is what the antibot does. It's basically growing up a bunch of viruses. I wonder if Dr. Delgado would like to have an antibot, but she could use it for her own viruses. I don't know. It's pretty specific to phage display. But, I'm sure she wouldn't turn it down. So the thing is, what this does is this infects bacteria with viruses, and it uses an antigen, the antigen extracellular domain. They put the whatever the surface protein is on, and they connect it to an FC domain that they use as part of the technique. Now, how exactly they do this is kind of too biochemical. I'm going to leave you to actually look at it, but I want to tell you what you need to know about this. You need to know that it uses the antibody FC domain the same way that our cell uses an antibody FC domain, and then it puts the antigen on streptavidin beads. So basically the biotin sticks to streptavidin beads. It immobilizes the antigen. See that on the right? Immobilized antigen. So the antigen is stuck to immobilized on a bead. Then you flow a phage library over it. What you've done is you've taken a bunch of bacteriophages with a bunch of antibody sequences on them, and then you've mutated the antigen binding parts of those sequences. You've mutated the variable regions. In fact, specifically you've mutated the CDRs and the HV1, 2, 3. Exactly what we talked about. So the thing you don't do gene rearrangement. You do a sort of supersomatic hypermutation to be able to mutate just the loops of the protein, and then you have like millions of those. You flow the millions of bacteriophages over the immobilized antigen, and some of them stick. When they stick, you wash the other ones off, then you elute the ones that stuck and you sequence their DNA or RNA, whichever one they have, and then you read it off and you know what the antibody sequence is, what the mutation was that successfully bound the antigen. The cool thing about this is you can actually do it in a circle. See, on the right you have a circle where you bind, you sequence, you pick a sequence, you select, and then you can vary that sequence again. You can do several rounds of phage display, and this is like somatic hypermutation as well. You can pick particular residues, you can mutate them, make a whole bunch of variation of just those residues, then flow them all over and see which ones stick. You're literally throwing them all at the antigen and seeing what sticks. And it's because bacteriophage has genetic material that you're able to just read that genetic material and find out what has stuck. It's very cool. This is how they describe it in the paper, but I described it in totally different words because I want you to get the concept. One other thing, this is not in the paper, but this is in the talk that Wells gave. Why did they use that particular FAB fragment? Why did they choose that sequence? Well, they actually chose an old scaffold that expresses very well. So they don't do gene segment recombination. What they did is a different kind of selection, because they did a bunch of experiments way back in the day when Genentech was being founded. They found that there was this one FAB sequence that bacteria like to make. Bacteriophage made really well. And they don't know why, but they just know that it makes a stable, highly expressed, high amount of protein. And so they use that as their scaffold. And so there is a selection that happens there as well. I think that's the kind of thing that you don't get in the paper. In fact, you don't get much of the antibody in this paper at all. There's some cool biochemistry going on, but I'm going to leave it at this. I think the phage display concept is what you need to get, because that's parallel to B cell variation. Okay. So there is a separate article that talks about how the antibody comes in, because that's not in the paper. And the antibody uses phage display. This is where it says the antibody uses phage display. This is another view of phage display. So I know it's a lot to take in, but it's the key concept that you want to see right here. And it's a cycle where you select for binding and then you amplify what's bound. Then you select again. You vary, select, amplify. And what you do is you literally, whatever sticks to the target is what you choose to amplify. Because the bacteriophages infect E. coli, you infect the E. coli with the bacteriophage and you amplify the sequence that you need. Okay? So that's very cool. You can look at this, and the one thing I want you to see right here is possible to perform selections on antigens in a 96-well format. If you look carefully at the antibot video, you will see 96-well plates being moved around by it. This is what the antibot is doing. So the only question is, where are they varying the antibody? Because you can't vary every residue, not to mention you don't need to. You just vary the residues on the variable loops where they contact antigen. And this is where they tell you exactly how they even build the DNA that results in the variation that they're talking about. So look at what they're doing. Look at the bottom down here, part C. They are varying individual amino acids. You can see that there's not that many amino acids that they're varying because they just need to vary the amino acids that will contact antigen. And they're actually just varying 1, 2, 3, 4 of the CDRs, of 6 possible CDRs, and they just have to mutate 4 of them. Even that has a total possible diversity of 10 to the 30th. So technically they don't do a complete variation, but they don't need to. You just need enough variation to have a lot of diversity. They have 3 times 10 to the 10th sequences, and that is enough. That will get you something that will bind, and then you can select it, amplify it, and mutate that. So realize that I want you to see how this is like a robotic version of natural somatic hypermutation. It is not like gene shuffling. There's no gene recombination. It's all done on the same framework, and the only thing, this framework was selected by humans for its good properties in the lab. One more thing about this technical thing about this paper. They mention ELISA, and we really need to talk about what ELISA is, because we've talked about what antibodies can do in the body. Well, those antibodies can actually be used in the lab. And you might have seen this in a cell biology class. You need to get it in multiple classes. So here it is in immunology. ELISA is a particular thing, and if you look at this, ELISA is one of the things that the antibot is doing. They isolated 95 individual phage clones and screened them for binding by phage ELISA followed by DNA sequencing. ELISA is what's going on in those 96 well plates that you see in the antibot. And so if you want to see exactly how it works, if you're ever working on an antibot, you can look at this and we can talk about the different things that they see. There's some things I know Wells has researched well enough to know that he actually worked on protein A a long time ago. And look at protein A shows up here. Each scientist does things their own way. And one of the things that Wells does, if you had experiments that worked on protein A, you have a lot of it around the lab, and you build your antibot so it uses protein A. I just think that's kind of one of those things you can find out about science by looking at the details. The detail we care about is that the antibot was doing ELISA, and that uses antibodies. It's a way of using antibody to determine binding. And it's really nice because you can do it in a 96 well plate. That means you can do 96 experiments at once, or more likely like 7 experiments at once, given controls in different concentrations. Okay. So this is what ELISA is like, and I want you to notice how it's like the previous thing we have, where we have 2 antibodies working together. And it's really a binding assay. You immobilize your sample on the bottom. And for this sample, you have an antibody that binds the blue stuff. You want to know how much blue stuff there is. And so what you do is you put your whole sample down the bottom, and then you have some exposed parts at the bottom, so you cover those up with a nonspecific protein. It's actually oftentimes milk protein. That works very well, and it's not very immunogenic. So you incubate it with the primary antibody, which binds the blue antigen. You see, if you have blue antigen in the sample, it will bind, and it will be immobilized to the bottom of the cell. It'll be immunosorbed. That's one of the ways to talk about it. So then your primary antibody is just binding there. You need to see that it's there. So then you add a secondary antibody that has an antibody enzyme complex. It has some way to see what it is. The secondary antibody binds the Fc of the primary antibody. That's the key idea here. You want to see that. Secondary antibody binds, then it has an enzyme with it, and all the enzyme does is it turns some substrate from one color into another. You add the substrate, it makes the red product, and wherever you see the red product, you know you have the enzyme. Where you have the enzyme, you know you have the secondary antibody. Where you have the secondary antibody, you know you have the primary antibody. Wherever you have the primary antibody, you know you have the blue stuff. So you look for the colored molecule at the top, and it tells you have the blue stuff at the bottom. Now this is big business. There's some devices that will do this for you. These are the regular lab devices that will do ELISA for you, the different steps. And the only thing I want to say is these were automated and put in a robot, so that can run 24/7. But it still needs humans to run it, to set it up, and to fix it when it breaks. And this is what the results look like. The ELISA assay, the 96 well plate, is shown on the left, and you can see whatever the antigen was that the primary antibody was binding, the yellow spots have more of it. And so you do 96 experiments, and you put them all together. Usually you do experiments in a line. You draw a line, you see how much stuff you have. This tells you by the colors how much binding you have to the bottom of the plate. The other thing that you can use antibodies for is for immunoblots. This is another cell biology topic. But when you run a gel, you separate a whole mixture of proteins by their size. Smaller proteins at the bottom, bigger ones at the top. See how the numbers next to the gel, 97.4 at the top, 14.4 at the bottom. So the proteins are getting smaller as you go down the gel. Now if you just look at all the proteins, like let's look in, this is row 1, 2, and 3 are in order on both sides of the numbers there. And you can see in row 1 you've got a ton of different proteins there. You might know that your protein is about 60 kilodaltons big. And you see that in well 3 you have your purified protein that you're looking for. And so the question is that purified protein in well 1? Well, you see something at 60, but how do you know it's actually the protein that you think it is? Maybe it's something else that happens to be 60 kilodaltons. So if you look over at, so wells 4, 5, and 6 are blotted wells where the exact same thing on the SDS page gel was run, except that you blotted it with an antibody, and the antibody will stick to the gel in the exact place where that specific protein is. The antibody will bind to the antigen, and if the antibody binds, you know that that antigen is there. Therefore, you can see, oh, lane 4 does have the protein I'm interested in, and it's right at the right size, and it must be somewhere in that mess of proteins in lane 1. Lane 2 and lane 5 go together, lane 6 and lane 3 go together, and you can see that, if anything, the antibody binding in lanes 4, 5, and 6 is increasing as you move from lane 4 to lane 6. That means if your process is moving in that direction, that you are getting more and more pure protein as you do it. We're not going to do this, it's not a lab class, but you might see data like this sometimes, and you need to know that ELISA data means you have an antibody recognizing an antigen, and it's shown by color, either the color of a well or a color of a band in a blot at the exact location where that thing has been located. Western blots are really important. Hopefully you get the chance to do one in cell bio. But this is also a big topic. You can blot a gel by applying an antibody to it. There's a lot of factors. It's complex. How's a good antibody separated from a bad antibody? A bad antibody means it will just bind to anything. It will bind to more than one thing. A good antibody is very specifically binding to just one thing. And so you can see right here, Western blot bands. You can see that, like, these are what protein markers should look like. And like bad antibody right here in the middle, it's binding 4 different things. You really want to buy our antibodies from Thermo Fisher Scientific, is what it says at the bottom. The results from that will give you a single specific band, which means you know exactly where your protein is. You won't get bad antibody bands, you won't get frowny bands, you won't get felt tip markers. They're trying to make a joke, and they're trying to sell material. But there are big differences in how antibodies work. You don't know until you try them. So you actually do have, to bring us back to immunology topics, well, ELISA is an immunology topic because it runs on antibodies. And you can use them in proteomics. Like here, for example, is a big proteomics array where you have on the left, you have the serum from a U.S. adult who has never been infected by malaria. On the right, you have the serum from someone who is living in West Africa and has been infected by malaria. So they've got malaria parasite growing in them. It was thrown over a microarray. So what's immobilized here? They have 4,500 different proteins from the malaria parasite immobilized on this grid. They know exactly where each protein is. So when you put the serum over it, you actually will see it light up. It's like a big ELISA. And you'll see it light up at the spot where you have the antigen antibody binding, where you have that parasite protein present. And you can see on the left, you have somebody who's never been infected by malaria. They don't have any antibodies that bind malaria proteins. They have a couple here and there. The person who's been infected by malaria has a robust antibody response. You can see that maybe 1/8 of these proteins are generating some kind of antibody binding to them. And you can go in and see which ones. How does an individual make so many antibodies to this? But this is what somebody who's been chronically infected, their blood will be full with antibodies that will bind that malaria. Now in this case, malaria is so tricky, it's able to get away from the immune response. And part of that is it hangs out in red blood cells. More about that later as we get through the class to specific diseases. But I just want to say, this is just like the ELISA plate. What lights up shows you antibodies are binding to it. And so finally, if you ever need like a CRISPR Journal Club, you're talking about what CRISPR can do, they actually do a totally separate screen where they're using not mass spec, but they're using CRISPR. We're not going to get into that. This is just an immunology class, but I want you to know it's here because you've been introduced to this paper and you can bring it back and you can learn about it more. If you need to talk about CRISPR, consider this paper. The thing is, the CRISPR screen gave them the same results as their mass spec screen, their SILAC is what they called it. But they just gave them 2 proteins at the end. See right here, 2 genes were identified as hits in both experiments, ITGB5 and CDCP1. And CDCP1 actually has a history. It's been detected in a specific kind of cancer. So they went after it. Maybe it's involved in other cancers. And it's a nice correlation because other researchers found CDCP1 was important in cancer. They found it too, and it's worth going after. They don't say anything about ITGB5. Who knows? And maybe I should look up the literature to see if they found anything in the time since. But the main thing is, all the proteins on the surface of the cell were tested. 7 were made antibodies to, and 2 of them were also identified by the different kind of screen, and only 1 of them had previous literature to support it. So at what cost? And it's kind of the question. That's the whole question with immunotherapy. It's heavily expensive, and we have to ask these economic questions because in terms of equity and in terms of resources, is it really worth it to put all these resources into this when there are people dying of preventable diseases elsewhere that the resources could go to? Big question. Ethical question. And it does involve immunology. So it helps if you think about that. So I'll leave you with that philosophical point, that philosophical question, but now you know a lot more about immunology. You can read a really complicated paper, and I think that is definitely enough work for this week. So I will leave you with this, and I will see you in class on Monday.